This webinar, Do We Need to Change the Definition of Drug-like Properties, was recorded on Tuesday, the 21st of May, 2019. It's my great pleasure to introduce our speaker today, Mike Shultz. Mike is the heart failure lead at Novartis Institute for Biomedical Research. He joined Novartis in 2001 in the arthritis and bone metabolism department, moved to oncology disease area in 2004 based in Cambridge, Massachusetts, and spent a year in Shanghai at the China Novartis Institute for Biomedical Research. Then 3 years in the immuno-oncology group prior to joining the cardiovascular and metabolism disease area in 2017. He's led projects targeting HDACs, Tanqueray, IDO, and led chemistry groups in oncology, chemical biology, and heart failure. So a very wide range of experience in different target classes and therapeutic areas. His contributions to 4 compounds undergoing clinical trials helped to spark an interest in how metrics and properties are used during molecular optimizations. But I have to say, from a personal note, it's been a great pleasure to collaborate with Mike over many years now. I've always found him to be a very deep thinker about the definitions of drug-like properties and the impact that they have on decisions in drug discovery and the progress of drug discovery projects. One example I always like to cite is a very interesting lunch I once had with Mike. We were sitting there chatting, and he asked what on the face of it was a very simple rhetorical question. He just said, why is the drug likeness cutoff for hydrogen bond acceptors always higher than that for hydrogen bond donors? And I must admit, I'd never thought of that before. And in hindsight, it should have been absolutely obvious. But rather than me tell you, Mike, can I hand over to you, and can you enlighten us?
Yeah, absolutely. Thank you for having me today. Yeah, it was after having StarDrop in my hands for a little while, able to look at molecules and drugs and not drugs and take a look at them, that it really stood out that all the drugs had more hydrogen bond acceptors than donors. And I thought that was curious, and I talked to people, and it was always the discussion of desolvation or other explanations as to why they need to be permeable. And then I kind of just opened my organic textbook and realized that there aren't any organic functional groups that have more hydrogen bond donors than acceptors. So that's just a consequence of being an organic molecule. But thinking that that was a major drug likeness as opposed to just organic chemistry was not possible, or you could finally understand that after being able to look at large numbers of molecules through StarDrop. So.
Thanks, Mike. Well, we're going to hand over to you now and give you control of the presentation and take it from here. Thank you very much.
Great. Thank you for the invitation, and thanks to everybody for being able to attend and dial in today. Really excited to talk about this. It has received some attention, and I bring up this article that was written in the last fall, and being interviewed for one of the papers that we wrote recently. It struck me a little bit as to how it was being portrayed as perhaps maybe Novartis versus Pfizer, or looking at these ideas. And I think the one theme that I would like to get across today is just the scientific discourse that I think is needed, and some of the tools that we now have available to us today allows us to ask questions about things that we thought we knew. And I think we should continue to keep asking those questions. So as I get control here. So I think we're all familiar with the term drug-like properties, and these originally were started about 20 years ago with the Rule of 5 that came from Lipinski, Lombardo, Domini, and Feeney, where they examined a couple thousand U.S. applied name compounds, and they hypothesized that these molecules, which had achieved some level of oral exposure, could guide us into what would be required for a molecule to have oral exposure. And they found that by having this rule as the Rule of 5 based on these parameters that were sums of 5 or products of 5, such that hydrogen bond donors were greater than 5, clogP greater than 5, molecular weight greater than 500, and hydrogen bond acceptors greater than 10, that if you had two of these that were violated, you would have a much lower likelihood of being an oral compound. And at as is typical at the time of that paper, so the data was not included in that paper, nor was the supporting information. I think this is one thing that we've tried to do with some of our work to make sure that the paper and supplemental information contains this information for other people to examine it and come away with their own conclusions. So without that data, we could not go back and look at the data used to make those determinations. But since that time, additional parameters have been suggested, including polar surface area, such as topological polar surface area by Weber, and rotatable bonds, where Weber and co-workers correlated that with oral exposure based on their rat PK data. Fraction of sp3 carbons by Frank Lovering and co-workers, and Tim Ritchie and his colleagues came up with an aromatic ring count. So different ways of looking at parameters of molecules that would hopefully enable us to increase our chances of making molecules that were more drug-like. So if we just focus on the rule of 5 parameters, molecular weight, hydrogen bond donors, acceptors, and log P, we can see the distribution of these parameters are actually not a Gaussian distribution. And I've shown here in red the number of compounds that are excluded based on these cutoffs. And just looking at molecular weight, we see that it's a long tail. So 8.5% of the compounds were excluded by this criteria, with hydrogen bond donors about 5%, and slightly less than 10% based on log P and hydrogen bond acceptors. So these were the oral drugs approved by FDA through 1997. So I was able to retrieve this information and use this as an analysis, since the information from Lipinski was not included in their paper. I think most of us are familiar with the concept of hit-like, lead-like, and drug-like that has since followed. In comparison of the drug-lead pairs by several groups of people, found that in successful optimizations, molecules get bigger, more potent, and more lipophilic. And I think that's something that we've all taken as fact, and so this should not come as a surprise to anyone. Parola in 2010 did another analysis, and this is about the time when efficiency metrics were being examined, and her work was to see how efficiency metrics have changed, but her analysis did not actually find that molecules increase lipophilicity during successful optimizations. But from this work, the concept of lead-like from Aprea and the rule of 3 by Congreve and co-workers came in the early 2000s and has entered our concept of drug-likeness as well. So at Novartis, we were looking at where our clinical candidates came from, and we were beginning this work with the intention to look at where our screening deck was, and did we have the chemical equity needed to come up with the appropriate drugs. And so here are the rules that I've discussed previously, and the values for these rules. So lead-like, rule of 3, and rule of 5, and I included Weber's 10 and 140 values. In the nomenclature in this presentation, anything that's a hit or a lead will be called a start, and anything that is a development candidate or a drug will just be called an end. So here are the numbers, and there are a few tables that we'll be covering today, and so we don't really need to look too closely at the data, but Aprea, Proudfoot, and Han had similar values. They had varying degrees of, various sizes, I should say, of databases that they were looking at, but the numbers in general were quite consistent with one another. And so at Novartis, when we looked at our starting points, we found that for our optimization to development candidates, we found that the molecular weight and log P were a bit higher than what other people had reported in the literature. And when we looked at how our molecule started and ended up compared to the benchmarks that I just mentioned, we also found that molecular weight increased in about a similar magnitude as what had been previously reported, 50 to 80 molecular weight increase. Our log P went down slightly, although our ending point was roughly the same, and other parameters changed as well. But if we look at where our optimization starting points were, they were actually larger on average than the average drug of 2001. And our starting point for our log P was actually higher too. So Novartis internally, our internal examination suggested we were bringing molecules that were larger and more lipophilic into optimization than what the literature suggested was appropriate. So we went back and wanted to look at this a little bit further, and so taking the data for the FDA-approved oral drugs from 1900 to 1997 repeated the Lipinski hypothesis. So taking the 90th percentile cutoff of these parameters, and also comparing to the other papers, using the StarDrop platform as just a way to make it consistent across all these studies, we found that the 90th percentile for log-P, molecular weight, hydrogen bond donor, and hydrogen bond acceptors was about what Lipinski reported at the same time. So slight variation, and in their paper they said, we're trying to make a bit of a mnemonic to make it easy to remember. So there was nothing that was found different. So even though using a different data set, a similar conclusion was reached, and which now would now allow us to do a comparison over time. When we look at the median and mean values, we can see that the point where our ending compounds, our development candidates, were actually about the same as the 90th percentile cutoff that Lipinski found for drugs. And so if our starting points and our ending points are higher molecular weight than what was suggested acceptable, we, I think understandably, ask the question, should we be concerned? And one reason maybe perhaps we should be concerned is some of the work done by Wenlock and coworkers in 2003, where they looked at the number of FDA approvals over time, and they found that the number of FDA's approvals were declining. But if we looked at the molecular weight in the various phases of development, they appeared to also decline to a point where the values that reached the market. And their conclusion was that the attrition rate, the high attrition rate in clinical trials, was in part due to these molecules not having drug-like properties in terms of molecular weight. So very high molecular weight molecules in phase 1 did not appear to make it through to the market. So taking that together, we wanted to look at this a little bit further. So looking at the first decade after the Lipinski hypothesis was published, from 1998 to 2007, there's 195 oral drugs that were approved in that period, and it's about a quarter of all oral drugs that had ever been approved. And in the table that I'll show here and the next couple tables, what I'll show is on the top row the values that I'd shown earlier, so using the values of the parameters at the 90th percentile from 1900 to 1997, and then the 90th percentile below that is from this time period, the first decade after. We look at clogP, we see that there hasn't been any change in the 90th percentile cutoff for log-P. Molecular weight had an increase. The p-value for this is 0.003, so it would be statistically significant, as was the change for rotatable bonds, but perhaps not as substantial. And the other parameters didn't show a statistically significant change over this time period. Looking at the values for the average and 10th percentile sort of gives us a perspective of the range of these. And so in this reddish color, approximately red color, that is what I use to highlight molecules where these parameters, the values have increased. And I guess that actually shows the bias. Using red to show an increase would psychologically mean that we view this as being bad. But in the first decade after Lipinski, there was essentially no change over the distribution of log-P. Molecular weight across the board seemed to increase. No change in hydrogen bond donors. Maybe the average value of hydrogen bond acceptors increased a little bit, as did rotatable bonds. But in general, it appeared that molecular weight was the only parameter that increased across the board during this period. And the p-values are calculated two different ways, using the one-way ANOVA and also the Kerskel-Wallace, which is more applicable to a non-normal distribution, which, as I showed you earlier in the presentation, that the molecular weight is not behaving as a normal distribution. So looking at the most recent decade, at the 90th percentile cutoff, so we see that, again, molecular weight has increased even more substantially and more significantly, as has hydrogen bond acceptors, rotatable bonds, and the number of aromatic rings. One might argue that these are actually correlated to molecular weight. Interestingly, the fraction of sp3 has decreased. The 90th percentile has decreased over this period. And we look at log-P, the lower end seems to have increased significantly and substantially, as has a number of the other parameters. But one thing that stands out to me in this, at least, is hydrogen bond donors has been insulated from change. So across the board, the 2 decades since the Lipinski rule of 5, hydrogen bond donors, the distribution has not changed in oral drugs. Fraction of sp3 is the one parameter that behaves as if a parameter should, that if it was converging towards a value, so the 90th percentile decreased, the 10th percentile has increased, and the average hasn't changed. So that doesn't mean that this is a drug-like property. It's just sort of an interesting observation, whereas in general, these parameters overall tend to increase to varying degrees. So one of the comments that I had received in the manuscript was regarding how we were calculating logP, and then some of the feedback was clearly that, well, the star drop way of calculating it wasn't utilized by many other people, and the historical clogP parameters such as Pomona, Crippen, and Mocha, as is one that had been used in some companies as well, they would give different results. And so looking at the 90th percentile, so 3 of the logP methods for calculating logP gave a significant and substantial increase in the 90th percentile since the Rule 5 was published, but star drop did not, whereas all of them showed an increase for the mean and the 10th percentile. So looking at the experimental values, we see that for the reported experimental values, there has not been a significant or substantial increase in in any of the 90th percentile, 10th percentile, or the average value. So that's a little bit close in the average. And again, this might be, there's not that many. There's only about 30% of the molecules have reported logP values. But I think this highlights one aspect that's important, is that you can get different results for your logP depending on which method one uses to calculate it. But we found that at least with star drop, it was more accurate, or there was less deviation from the experimental values than with some of the other methods. So going back to this notion of declining productivity in the pharmaceutical industry, we can look at the number of oral approvals by the FDA since 1900, and in the early 1990s, there was the Prescription Drug User Fee Act, which, to help alleviate some of the backlogs in trying to generate drugs to treat HIV, the FDA now charge user fees such that the pharmaceutical companies would help pay for the trials and the approval process. And so this cleared a backlog of compounds around 94, 95, 96, where there was a tremendous spike as the backlog was cleared, and then it declined a little bit over time. So this notion that there's been an overall decline in productivity is really artificial based on that legislation that relieved the backlog. But over time, we see an increase of drugs, which over the last few years have set a record pace. So how have the distributions of molecular weight and logP changed over this period? So pre-Lipinski and in the most recent decade, we see that there's been a shift. So in molecular weight, the number of smaller molecules have decreased. The medium range between 3 and 500 molecular weight has remained relatively constant, and the oral drugs that have been approved over 500 molecular weight has more than doubled. With logP, we see that, again, the number of molecules approved on the lower end has shrunk, but it has been more with an increase in the median lipophilicity rather than on the high end, a subtle increase, but it's been a shift, not so much of a Gaussian distribution in terms of logP. So if we were to repeat the Lipinski hypothesis and say, well, let's look at just 1998 to 2017, so that's more than half of the FDA-approved oral drugs. For molecular weight, and we would get, of the original Rule of 5 parameters, we would get a different value for molecular weight, although the other parameters on the Rule of 5 had not changed. We do see that rotatable bonds, fraction sp3, and number of aromatic rings has changed. But again, looking at the average and lower end of the distributions, there has been substantial changes. Which begs the question, can a parameter truly be drug-like if that parameter changes over time? So if it really is drug-like, then that should not change over time. So if it's really a hard cutoff that limits permeation and solubility, that there should not be a time-dependent component. Surprisingly, what we found is the hydrogen bond donors was the one parameter that did not change in this analysis. So looking at this another way, so I take the data that I'd shown previously from Wenlock from 1993 and just flipped it around the other way, looking at the molecular weight, the average and 90th percentile molecular weight in reaching the market versus increasingly earlier stages of clinical development, and compared to the average and 90th percentile cutoff of molecular weight for drugs approved over various periods, what we can see is that the molecules have, with higher molecular weight, they weren't failing in the clinical trials, they were actually succeeding. They just hadn't had enough time to make it through the clinical trials and the regulatory approvals to reach the market yet. So over time, the actual increase in number of approved oral drugs has been primarily by an increased number of molecules with molecular weight greater than 500. And in fact, this isn't something that's recent. So if we were to look at these parameters over time, molecular weight has been increasing since as far back as we've been approving drugs. It's been a slow and steady increase over this time, and I use this red dotted line to show where the 90th percentile would have been if we drew it in, say, 1957, because it's not clear why 1997 should be the one year to use this 90th percentile cutoff, but it just shows that depending on when and where you draw the cutoff, it can be surpassed over time. So with regard to molecular weight and hydrogen bond acceptors, these values have increased and they do not appear to have reached a plateau yet. For polar surface area and rotatable bonds, there has also been an increase, perhaps not as fast or not as significant, but these have also been increasing over time. But more interestingly, logP and hydrogen bond donors have been parameters that have changed more slightly or more subtly over time. So lipophilicity and hydrogen bond donors appear to be more resistant to change over time. So we were looking at this in the context of where our development candidates were coming from, and at this time, Dean Brown and Jonas Bostrom published this really good article where recent small molecule clinical development candidates come from, and they were kind enough to share their data with us, and we obtained the SMILES for all the start and end pairs either via the supplementary information or directly from the authors from Hahn, Aprea, Proudfoot, and Brown, and our own internal analysis. And this gave us the ability to compare the start and end pairs. And so what we did is we removed all duplicates or any case where there was multiple starts that were reported to give a single end point or a single endpoint that gave multiple ends. So just for clarity, only single start end pairs were utilized. So each of these studies calculated these properties differently. And as I showed you in the clogP, that can make a difference. And so we recalculated using StarDrop to allow all these parameters to be compared directly. And we were asking the question, what has changed? So what is the average start versus the average end? And how have these changed? Which is a question that hasn't been asked too much, but I hope will become more clear a little bit later. Okay, so another table with a lot of data. And so it's grouped in the time frames from when these publications these data were published. So in the 2001-2002 period, the three papers I mentioned previously, those papers have roughly the same molecular weight, hydrogen bond donor, hydrogen bond acceptors, and clogP. And then over on this column here, I have averaged the total number of these. So we can see that the average of 2001, and with 2018, these data were pooled and then averaged, and that we can see that there has been a change in the starting points. So the hits and leads of 2001, 2002 have changed in the approximate 20 years, such that molecular weight has substantially increased, as has hydrogen bond acceptors and clogP, but hydrogen bond donors have not changed in terms of successful starting points and optimizations. I've included these other parameters that have been calculated, and I don't think I have much to say on these except for the fact that all of these have changed as well, which again sort of highlights the nature of hydrogen bond donors not changing as being different from the rest of these parameters. So comparing the starting and the end pairs, again, as I showed you previously, the starting points have changed significantly. Looking at the end points in a similar analysis, they have become larger hydrogen bond donors have not changed that much. Hydrogen bond acceptors and logP have all increased for the ending points, and that we can see that in all cases starting and ending points, there is an increase in molecular weight, hydrogen bond acceptors, and clogP on average for successful optimizations. And this has been in line with where the parameters for approved oral drugs has been going. So the changes in hits, leads, and drugs have been fairly consistent over time. And again, to complete the very large table, lots of data that I'm sharing with you today, we see that for a lot of these other parameters, again, similar increases over time, both with starts and the ending points. So hits, leads, drugs, and development candidates are all changing in all of these parameters with the exception of hydrogen bond donors. This really begs the question, is this really pedantic or is this important? So going through all these numbers and nitpicking over what numbers have changed and which ones haven't, why is that actually important? So an increasing majority of hits and leads are no longer lead-like, just like an increasing majority of drugs are no longer drug-like based on these parameters. So what I show here is a molecular weight of the starting point. So pooled all of these the molecules from 2018 and the other ones that I mentioned earlier. And so we can see with the molecular weight, the distribution of molecular weight and clogP. So one of the things that I like is that to point out is logP is really the only calculated property. All the other parameters are descriptors. So molecular weight is known with absolute precision for these molecules, and it's not actually a property of the molecule, it's just a parameter that we calculate. And that we can see with logP, this is a calculated property that is associated with an error in the calculation, and there's an association with the error in measurement, whereas that does not apply to the other parameters. This highlights that when we use these absolute cutoffs that we can see that there is actually a difference between these two values in terms of their molecular weight that we can with certainty say they're different. Whether they're substantial, that is hard to say. But in terms of these molecules here, where we see the error bars with these whiskers here, the difference between logP is too close to be outside the error of calculation or measurement. So somehow some of these molecules are acceptable and some of the other ones right around it are not. But these are acceptable to the same degree that these molecules are. Where it gets even more interesting, in my opinion, is when we look at the rule of three. So 85% of the modern starts are not rule of three compliant, mainly because hydrogen bond acceptors are extremely punishing to these molecules. So only 15% of modern starts are rule of three compliant. This is important if any company is using these values as a triage mechanism for high throughput screening or how to organize screening decks or how to look at molecules, how to select molecules. And so this is just the point of caution with these. So over time, the properties hits, leads, and drugs have changed significantly. There's been statistically significant and substantial changes to the 90th percentile, the rule of five cutoffs for oral drugs approved by the FDA for molecular weight, hydrogen bond acceptors, and rotatable bonds. There have not been any change in hydrogen bond donors for hits, leads, or drugs. And although the average logP has increased for hits, leads, and drugs, the 90th percentile remains about 5, a more subtle increase over time. So now we want to look at how these parameters have changed. So that was the what has changed, but how have they changed? So most of the publications that are out there, all the ones that I could find, they look at average values. And the question that we asked is, well, do all the values change the same? Or perhaps could the changes be dependent on the starting point? So with all of these data, as I mentioned previously, with the match starting and end pair, we were now able to ask these questions. So during successful optimizations from the data that we have been able to obtain, that although there's an average increase in molecular weight, it depends on where your molecular weight of your starting point is as to the magnitude of change in your molecular weight. That is that molecules that are smaller, so the smaller starts get much larger, whereas large molecules tend not to increase in molecular weight. So this is important because if we're using criteria that we say on average molecules all get larger, if that's the case, we don't want to start at a 500 molecular weight molecule because it will also get larger. That has not been borne out by the data that has been published or that we've been able to analyze. So smaller molecules get much larger, medium molecules get a little bit larger, and on average, large molecules tend not to change molecular weight substantially. Now, depending on where one starts off in terms of calculated log P, the similar effect on the ending point occurs as well. So I apologize, this didn't come through in the slides, that this is the C log P range. So this should be -2 to -1, -3 to -2. Whether we're looking at just 2018 molecules, so the Brown and Novartis data, or all of the data, we found that molecules with very low C log Ps tend to increase log P during optimizations, and molecules with large log Ps tend to decrease log P during optimization. And within a certain range, they remain relatively unchanged. So again, for these two parameters, the low end tends to get much larger, the high end tends to stay the same or decrease. Interestingly, during successful optimizations, that molecular size is added with an increasing amount of hydrogen bond acceptors, but not with hydrogen bond donors. So during an optimization, the change in molecular weight is shown on the x-axis here, and the change in hydrogen bond acceptors on the y-axis that we see as molecular weight is added, it's done via hydrogen bond acceptors and not so much by hydrogen bond donors. So that on average, an increase of 300 molecular weight during an optimization is accompanied by 4 hydrogen bond acceptors being added, whereas that 300 molecular weight increase is accompanied by a half of a hydrogen bond donor during the same. So about an 8-fold higher rate of hydrogen bond acceptors than donors based on molecular weight. Looking at topological polar surface area, these values tend to converge. So again, molecules with the low polarity tend to get more polar. In that middle range, molecules they tend to change very little, perhaps get slightly more polar, and molecules that have very high polarity tend to get less polar during successful optimizations. So this is a table that tried probably against my better nature to sort of quantify it, is depending on where your starting point is, what kind of magnitude has occurred in the literature. Again, I want to be careful that these should not be rules, that we've already seen that how things have changed in the past don't necessarily predict how they will in the future. But more or less, this is just to say that even though the average of these parameters might change to a certain degree, it is dependent on what that starting point is for your molecule. So this might be useful depending on what you have to kind of predict what needs to change. So if you're on the high end of molecular weight, you wouldn't expect it to get much larger. So let's just think about this for a moment and how we derived some of these rules. So this is a variation of the Hitchhiker's Guide to the Galaxy. Imre Mahak has this quote. He used Douglas Adams' quote about chemical space. And chemical space is really big. You won't believe just how vastly, hugely, mind-bogglingly big it is. I mean, you may think there's a lot of variation in a phage or Dell library, but that's just peanuts to chemical space. So let's put this into perspective. And I think we've seen some of these numbers I'll be talking about in a minute. But what we've shown previously is looking at the data we have. So looking at the properties of drugs, hits, and leads to tell us what drug likeness is, but that is committing the base rate fallacy. So that means that we have to look at not drugs and compare them to drugs to determine if there's actually a difference, because maybe drugs just look like the statistical distribution of molecules in a corporate archive. So in order to do this, so the paper this published fairly recently where heavy atom count up to 17, the GDB 17, they enumerated 166 billion organic molecules. And so that's still in the fragment space. We're just getting out of fragment space perhaps. That's significantly more than the number of FDA-approved oral drugs. And if you look at the CPU time to calculate what they were able to calculate, if we got up to 30 heavy atoms there isn't actually enough CPU time in the age of the Earth or the universe to have actually calculated all the non-drugs, and this would still include molecules that are important to companies such as mine, such as Gleevec. A lot of drugs would be excluded. We can't even come close to calculating what non-drugs are. So to compare between drugs and not drugs, there just isn't, we do not have the ability to do this. So the estimated number of drug-like molecules has been put at about 10 to the 63rd. I know there's varying estimates out there, sometimes higher, but this one is, again, heavy atom count less than 30, hydrogen, carbon, nitrogen, oxygen, sulfur, but no stereochemistry or no halogens in these molecules. So very clearly a conservative estimate of the number of drug-like molecules. The typical large company corporate screening deck is on the order of 1 million molecules in the screening deck. The CAS registry has about 100 million molecules. The largest single collection of molecules that has been reported that I'm aware of is the New Evolution DEL library, which is about 40 trillion molecules. And if we put this into perspective, the age of the universe is about 10 to the 17 seconds. If we were to synthesize a New Evolution DEL library every second since the beginning of time, we still would not be able to cover the drug-like space conservatively estimated up here. We'd be well short of that. And if we wanted to do something, there's actually not enough matter on the planet to make one of every drug-like molecules that could exist. Looking at the molecules used to derive the rule of 5, this is substantially smaller, so about 10 to the 3rd number of molecules. And the fraction of the drug-like space used to calculate what drug-like is, one can see there's quite a bit of decimal places associated with this number. So the concept of drug likeness, as we've been looking at it, has changed over time. We can't look at the non-drugs, and the number of drugs used to predict all of drug-like space is a very tiny fraction of what the possible drugs could be. So to summarize this, the rule of 5 was really implemented at a time when experimental permeability and solubility were very hard to obtain, and the predicted values were unreliable. Things are different today. We have much greater access to experimental solubility and permeability data, and remember that the rule of 5 was only for solubility and permeation. So we can measure and predict these for lots more molecules today. So we do not need to use the rules that were developed previously to predict that. We have much better predictive tools. With the exception of hydrogen bond donors, the properties of hits, leads, candidates, and drugs have continually changed over time. So this is not a recent phenomenon. They have been changing since the beginning of time. One question that I often get is, why is this happening? And one of my favorite quotes by Stephen Wright, a comedian, he says that everywhere is walking distance if you have the time. That's the same thing with molecular weight. It's really, in my opinion, is a reflection of synthetic accessibility. You can make a molecule as large as you want if you're willing to put enough time into it. And as I stated previously about hydrogen bond donors, not really changing much during optimizations, and as we said earlier, hit-like, lead-like, and drug-like properties, they either do not exist or we have not yet reached the plateau in these parameters needed to define them. So I'm just going to end by calling out the Wright brothers and their horrible model for commercial airlines. Okay, so I like to use this analogy because if it's perceived that there's criticisms of Lipinski or the rule of 5, that would be exactly like me saying the Wright brothers had really a horrible model for commercial aviation because, I mean, just look at the seating here. There's one person laying down. That's not a very comfortable flight. They crashed 50% of the time on this flight, which is a terrible safety record. There is no frequent flyer miles associated with this. The baggage allowance was horrible. Their on-time schedule was terrible. And we can go on and on about how the Wright brothers was a horrible model. And this can be applied in one way to sort of the rule of 5 and say, with hindsight, we can say we've advanced so far and that model should not be used. We should recognize it as the advancement in the field that enabled this concept of drug likeness and a lot of us to consider it. And we can hold it high as saying it was a major advancement that also deserves to be put in a museum and no longer used in our day-to-day decision making. So I opened with that article and ended here with the point saying that I believe we should be able to look at the rules and our concepts that we use in drug discovery, but we also need to do, and we can do so critically, but it doesn't mean that it was a bad idea at the time or it wasn't a major advancement for our field. So all that remains is for me to thank everyone for your attention during this webinar, and the people that made this analysis possible. And some of their names are highlighted here as part of the team where we looked at our starting and ending points internally at Novartis, in addition to all the chemists at Novartis who really helped enter the data needed to make this analysis possible. And then again, I'd like to thank Matt, John, and all of Uptibrium for the opportunity to share some of the findings today. And I'd be happy to take any other questions.
Hi, Mike. Thanks a lot for a really fascinating and very detailed analysis. It's great to see StarDrop put to such great use. We can already see here that there's a number of questions already coming in. But just to give the rest of the audience a chance to type in their questions, I just thought it'd be really interesting. You shared a really funny sort of anecdote about some of the feedback you had from the reviewers of your paper in JMed Chem, and particularly about, as you mentioned, the choice of the StarDrop clogP model to do this analysis. I wonder if you could just share that with the audience while we sort out all of the technical details here.
Yeah, absolutely. I mean, so as many know that in any article goes through multiple iterations, and certainly the reviewers were instrumental in improving the final product. You know, in sense, rightly so, I was using the StarDrop model, and many of them hadn't seen it before, and so there was questions about it. Why aren't we using the old models, and how are they different? And so that was the process where I had to go back and then pull up the data, go through, recalculate via a variety of parameters. And honestly, myself, I hadn't done that previously, and I was a little bit worried. So maybe I came away with entirely incorrect conclusions based on this. But I think the point that the properties have changed, perhaps the cutoff hasn't, depending on which model you use or if you use experimental data, but it just really highlights that when people use the term clogP, it could be highly dependent on which model that we use. So in our analysis, it looked like, whereas the other models seemed to substantially overpredict clogP, StarDrop had a subtle underprediction for the very high clogP molecules, which is arguable how well we can measure those anyway. But
thanks very much, Mike. We've got the questions are starting to flood in. The first question is, do you think that ligand efficiency would follow a similar growth trend to molecular weight? Or is the growing molecular weight perhaps reflective of higher affinities or better understood mechanisms and targets?
So with regard to ligand efficiency, I have a few strong opinions on that metric, and mainly I think it had been shown previously by several groups that it's not a particularly useful or accurate metric for comparing molecules of different sizes. So at higher molecular weights, it just swamps out all aspects of it. I don't have the affinities for the drugs listed. I think some of them are available. I intentionally included as much data as possible in the supplementary information because I think that's an interesting question. I think other people that, you know, they may have a different opinion of that, and they may then use the data hopefully to ask those questions. So I've not done that detailed analysis, and I'm just trying to be upfront about the bias that I have. Maybe somebody else can do that analysis and gets a different conclusion.
Thanks, Mike. All right, the next question, it says, small starts get larger and large starts do not. But is this biased to the screen that's being performed or a particular target?
Excellent question, and I wish I had an excellent answer, but I do not think I do. We have, for our internal data, we have the screening method to identify, that tells us how we identified our starting points. We don't have that information for the published data. So our data set being much smaller, it would be hard to do that. I think it's tempting to to look at. I think there's been a lot of good papers recently about fragment-based drug discovery. It may also come down to synthetic ease. You know, in a very large hit, it's going to be prohibitively the time is going to be prohibitively expensive to elaborate and grow a large molecule very, very much larger. With a small molecule, maybe a couple of them can be linked together, so to speak. But yeah, I could see how the screening methodology would influence that.
Great, thanks again. Okay, so the next question is very related. It says, should we alter our screening methods and libraries to accommodate not having to change parameters so drastically?
That's a good question. So we're in the process of doing away with molecular weight and log-Ps as cutoffs for what we use. I think because we're, as an industry, we're able to predict solubility and permeability much better. And we know, and we have a much better sense of whether our predictions are whether we're confident in those predictions. Perhaps we can choose those molecules. Personally, all things equal, if I have a couple hits, and there's a program that we're doing this now, is that we got multiple hits, I'm going to want to use the smallest molecule possible, just because I know the cycle time for optimization will be faster. But that being said, one never knows with a new target what chemical space is going to be needed to modulate that target. So I would tend to be against altering the screening decks based on size, unless one had really good information on why to do so.
Fantastic. Okay, so in considering how the parameters change, do you need to take into account the number of examples in each bucket? Have you taken this into account?
Maybe Clint, is the question into each bucket save the molecular weight bin?
Yeah, so my assumption is when you're looking at parameters changing for the different sort of starting molecular weights, for example, the different bins, have you sort of taken into account the statistical significance in terms of the number of compounds in that bin?
I don't think I have. It was a very rough calculation, and certainly in the middle of those distributions are where the data is more heavily centered. Certainly a log P is probably the closest to a Gaussian distribution that we have, but on the ends you do see a tilt on that. You know, so even though there's fewer examples on the fringes, that the low log Ps almost always go higher and the high ones almost always go lower, and it sort of moves towards the center on that. That would sort of argue against. I think if they all changed to the same extent, then one would see that fairly easily as well. And so that was on that analysis that the thing that stood out and surprised me the most, because I wasn't expecting that.
Thanks, Mike. But if I'm right, just to confirm, in your sort of analysis of the changes and the averages over time for drugs and starts and leads, you had the statistical significance in each case. So that was very much tested rigorously with the appropriate sample sizes.
For the rule of five, so for the FDA approved oral drugs, we have done that. For the starting to ending points, that is still in the process of being written up, and that statistical analysis has not been done.
Okay, great. Thanks a lot. Oh, okay, we've got one asking for a forward-looking statement. Do you anticipate that we're heading towards a phys-chem plateau in oral space? And if so, where do you estimate it to be?
Well, I think there's been a couple really good papers over the last couple of years, and I tried to cite a few of those, and there's been subsequent ones as well, that talk about where, to the best of our ability, we can see such molecular weight that would limit oral absorption of permeability, and it seems to be about 950, 975, I think the estimates are, that when you get beyond that size, the molecules are too large. There's also a more concept that I have seen and we've tried to use in our design internally, and that's the chameleonic properties. So if you have a molecule that's of a larger size and it can change its conformation, one can imagine, say, cyclic peptides that it can rotate or it can modulate its conformation to bury hydrogen bonds, to cross a membrane, and then expose them when it's on the other side to increase its solubility. So those were sort of the molecules that might have been the exceptions to the rules when they were generated, but maybe they actually highlight what's important. So small molecules they can't do that, So on a very small molecule, if you have a zwitterionic compound, they won't be able to typically come in contact with each other to improve permeation. But a larger molecule, you can get a zwitterionic compound where the ion pair then comes in close contact, where it can then cross membranes, etc. So I have seen that the papers with the data supporting at that molecular weight at a certain level would be a cutoff for things like logP. I think it's questionable at a high enough value whether it's actually meaningful or measurable. But there are opportunities for larger molecules that don't exist for smaller molecules, such as the chameleonic properties. And so that's where I would say, I think, where, if I was to be forward-looking, I'd say that's where we will get much better in the future, is our ability to understand that and incorporate that in our design.
Thanks, Mike. Okay, so gosh, they're still coming in. So the next one is it being suggested that property-activity relationships should not be tracked during optimizations?
No. I think it's important, but to me it seems hard to a priori know exactly where you need to end up. I think it's important to monitor them and to understand these. Certainly in some optimizations that I've been part of and observed, that we can identify liabilities, and you might have a hErg or microsomal or permeability, et cetera, things that have been shown to be pretty strongly correlated with, or reasonably correlated, maybe I should say, with something like lipophilicity, et cetera. And so monitoring that along the way might tell you, all right, here's ways we can modulate this property while maintaining the activity that we want. What I've cautioned against is some people in my group are when people say, well, we want to make our compounds more bioavailable, increase oral exposure, so we're going to decrease molecular weight. So things that aren't related to the outcome that somebody wants, then those are the ones that shouldn't be used. And I think that was part of where this concept of drug likeness could be sort of harmful, is that we could be optimizing for things that are irrelevant for the outcome that we really wanted. So perhaps if one could show, maybe within a certain series, that some of these parameters do correlate with outcomes, I would suggest looking at it, and if you find that that's the case, then certainly optimize for it.
Fantastic. Well, the next one is, the questioner says, I agree with the notion that permeability and solubility are fairly predictable, but toxicity is much harder. Various people have shown that toxicity seems to track with some of the properties of lipophilic drugs, for example, aromatic ring count. What do you think?
So I think there's been several good papers showing that. I think there have been a couple other papers recently too showing that the data set used to determine some of these rules on certain parameters and as how they relate to toxicity don't replicate, or you get different outcomes depending on the different databases. And so there has been attempts to pool some databases, and certainly things like lipophilicity have tracked quite well, and that would seem to also correlate with aromatic ring count in general. That the more aromatic rings that are present in a molecule, they tend to be more lipophilic. Whether it's the one or the other that's driving it more significantly, that I don't know. But I do believe that the aromatic ring count, it's fairly convincing that does trend with toxicities.
Fantastic. I'm aware that we're sort of coming to the end of our allotted hour, but we've still got a few questions, and Mike has kindly volunteered to stay on a little bit longer. So we're going to keep going just for a few more minutes and get a few of the sort of high priority questions. and we'll try to get to them as many as we can in the next few minutes. And so the next question is actually related to the idea of prediction. And this question is, to what extent is a company like Novartis using predictive models of, for example, metabolism, protein binding, solubility, etc., in its optimization of hits to leads, or even in constructing libraries for initial hit screening?
So I think, like everybody, we're interested in doing this, and any point that we can eliminate the expensive experimental data gathering for accurate predictive tools, then we certainly want to do that. And I think most people who are or have been at a large company, it's typically the case that the entire company doesn't follow a single path. There's a range of views. But certainly we're encouraged and to a large degree, and I certainly am an advocate of this, is if, for example, if there's a metabolism issue, is to test the models, and if they're predicting well, then use them. If they're not predicting well, then not to use them. But we, don't assume that they're always going to predict. Get some data, and if the data confirms it, then rely much more heavily on the models.
Fantastic. So I'm going to group together a couple of questions here because I think they're closely related. So one asks if these trends might be different for different target classes, and the other says, are these trends or trajectories different for phenotypic projects compared with those for target-based approaches?
So I think the questions sort of hit on a part that I think is the most important aspect. I think we all recognize that the not only has the synthetic capabilities changed over time, so we're able to make things that we weren't before. I mean, I've been at Novartis almost 20 years, and sometimes I see the kind of synthetic work that we're doing, and I'm just thinking about as to when I started, saying, wow, what a change, what things we can access, chiral HPLC, et cetera, et cetera. But perhaps even more dramatically is the biological space that we're going after has changed. So that's where part of the notion of not wanting the old rules to get in the way of new modalities, until we test out protein degradation or other new modalities, assuming that they can't be an oral drug might be doing us a very strong disservice. So I'm familiar with a lot of the literature that shows that, and I think we can see this, different target classes have much different preferences for the kind of substrates or molecules that can modulate them. And so that's in part where going into new areas, we shouldn't be constrained by perhaps what kinases did or GPCRs how they might have behaved if we're going after, a brand new target class. So yes, the biological space we're going after has changed, and it impacts the molecules that we make. They have to be paired or made, bespoke for those classes. But that's probably about the best answer that I could give. I don't know exactly how much of it they've changed, but they certainly have.
Well, again, thank you very much. So I think we're just going to have one more question before we wrap up. My apologies to those who we didn't get round to, but the last question for today is, the ability to calculate parameters has improved, but so has the ability to deliver them with advanced formulations. How should this be accounted for in this analysis?
That's a great question, and this gets brought up a lot. How it gets accounted for in the analysis, again, this is a part where that's beyond the scope of the work that I did, but the information's there, and that would be extremely interesting for somebody who has the expertise that could say, oh, we are now seeing this is where formulation works or where it does not. I mean, we certainly don't want to be in a position where we're relying too heavily on formulation or just thinking anything's possible because of some of the heroic formulations. But yeah, it's clearly important. I just do not have the ability to say the magnitude of impact that the formulations have had.
Well, thanks again, Mike, for a great presentation, a very sort of insightful analysis, and for answering all the many questions that have come in today. I'd like to say a big thanks to all of our audience for joining us. It's been a great pleasure, and thank you for all your questions. And we'll look forward to speaking to you again soon.